Hello dear student, welcome to the journey of microbiology. Today we will start with the introduction to the medical and clinical microbiology. Myself Dr. Jigar Gusani, I am MD microbiologist. Let us begin. So, what are the objectives of today's session? We will learn what is microbiology. Then we will see what is medical microbiology and its sections. What is clinical microbiology and the importance of clinical microbiology in healthcare. So what is microbiology? Microbiology is basically study of microbes. Now these microbes, they can't be seen with naked eyes. They cannot be seen with the naked eyes. And so we require the help of microscope under which we can see them. Okay. Now microbes are everywhere on the earth. They are the indispensable part of ecosystem of the earth, and they outnumber any living species put together. More than 95% of microbes are harmless to us. In fact, many are beneficial to us in direct or indirect way. So the microbiology is very vast science and very huge science. In healthcare, what we deal with is the medical microbiology. So what is medical microbiology? Medical microbiology is the discipline of microbiology which deals with the study of pathogens. So what are pathogens? These are microbes which can cause infections and disease ultimately in humans. So they are potentially dangerous microorganisms which can cause disease in the human being. Broadly, they are categorized into bacteria, fungi, viruses, parasites, and other agents like prions, which cannot be categorized under these headings. Now, the study of bacteria is known as bacteriology. The study of fungi is known as mycology. The study of viruses is known as virology, and same way the study of parasite is known as parasitology. Now we have seen in the history as well as in the current time that world has gone through devastating pandemics and epidemics, and it is continued as we are currently going through the COVID pandemic, right? So studying pathogens is very important, and then we can find out the solution to fight against them, right? So in medical microbiology, basically what we do is we study pathogens. Okay, we study pathogens. So what do we study? Their morphology, their culture characteristic, their virulence factor, all virulence factor, right? The pathogenesis in general, then the host uh, defense and clinical manifestations. They produce in general, including various systems. So basically here the pathogen is at the center and it is being studied in detail so that its morphology, culture characteristic, virulence factors, and what kind of damage it can cause into the host, how it enters into the host, and what type of different clinical manifestation it causes when it enters into the host. Now let us see what is clinical microbiology. So clinical microbiology is an application of medical microbiology for the management of the infectious disease. So we are applying the knowledge of microorganisms to manage various infectious disease. So let us see which are the components of the clinical microbiology. So the first component is syndromic diagnosis of infectious conditions. This means that patient comes to us with a signs and symptoms, and these needs to be categorized. Say for example, a patient presents with the acute diarrhea, or the patient comes with a diarrhea along with blood and mucus, so we will call it as a dysentery. Or the patient comes with the signs and symptoms of neck rigidity and CNS involvement, so that will be under acute meningitis. Once we have identified a broad syndromic category, then based on the knowledge of various microorganisms, their characteristic and type of clinical manifestation and patient history, we will order certain tests and then we will identify the pathogen and then we will proceed for the treatment. So the first component is identification of syndrome and the signs and symptoms of the patient. The second step is obtaining appropriate samples and selection of appropriate tests. Now remember that the journey starts with the collection of samples, and it is very important in microbiology. Also, it may be possible that for one infectious disease there will be many tests, many diagnostic tests which are available. However, it is the knowledge of clinical microbiology which will help the clinician as well as laboratory medicine doctor that which tests are to be selected based on the signs and symptoms of the patient. So let us see what kind of tests are available. They are broadly categorized into these three headings, that once the samples are collected, they can be proceed with the direct microscopy. So here the samples are processed with either wet mount or staining and then seen under microscope. So various microscope based techniques are done here on the direct samples, and many a times we may be able to identify the pathogen easily, and it's a faster process, very quick result, right? The next step is cultivation and identification of the microorganisms. So here we grow the bacteria or the viruses or the fungi in the lab, and then we, after they have grown in the lab, we process them with various biochemical reactions or the biochemical tests, so that we can identify the pathogen and its species. Okay, the third category of tests, it could be when there is no direct observation in the microscope, there is nothing seen in the direct microscopy, or there are certain microorganisms which cannot be easily cultivated and their identification becomes very difficult. In those conditions we go for the indirect evidence. These include serological based tests, where we detect presence of antibody or antigen in the blood of the patient. So this gives indirect evidence of presence of microorganisms. Also, there are various advanced and automated tests. One of the example is molecular test like RT-PCR. So these are the broad categories of tests which are available, and the knowledge of clinical microbiology will help the clinician to select appropriate sample as well as proper tests. The third portion or the third part of clinical microbiology is infectious disease pathogenesis. So knowledge of pathogenesis means how the microorganism enter into the body, how it attaches to the various specific cells or the organs, and based on the virulence of microorganisms and the host response, the microorganism may be able to colonize, it stays there for a longer period, and it may be able to invade deeper tissues. Okay. Also, we will study in the pathogenesis how the host will respond, how its immune system will respond and counter the microorganisms. Also, it may be possible that microorganisms will counteract these health host responses with various evasion techniques or the mechanism, and they may be able to survive in the host and they may be able to multiply. Ultimately, if the pathogen is powerful, it will lead to tissue damage, and that is known as infectious disease. Remember that there is difference between infection. Infection means entry and attachment of the pathogen, but disease means it leads to tissue damage. Okay. The next major component or very crucial component of clinical microbiology, that it helps in guiding the antimicrobial therapy. So these will start with the identification of the microorganisms. So if we know that this is cocci or bacilli, and whether they are gram positive or gram negative, we can select an empirical therapy or empirical antibiotics, because the antibiotics will differ in case of gram positive and gram negative. Next, uh, we do with various tests like antimicrobial susceptibility tests, right? Or we can do molecular characterization for the resistance gene. This will give us the view that which antibiotic will work and which will not be work in the patient treatment. So ultimately it provides an invaluable local microbial data, what kind of microorganisms are growing in our hospital and what kind of resistant pattern they are showing, will help the clinician, will guides the clinician for starting empirical therapy as well as modifying the whole treatment. Once the results of these tests are available, they can modify the treatment. Okay, now the final and major component of our healthcare system should be always prevention of disease. That should be the highest motto of any physician. So clinical microbiology also helps in the prevention of various infectious conditions by these three components. So it helps in development of various vaccines. So vaccines are very helpful for immunoprophylaxis. Second component is hospital infection prevention and control. So hospital environment is prone for various cross infections, and we need to prevent our staff as well as patient in getting the new infections from the hospitals. The third is public health measures. So clinical microbiology along with the epidemiological study. So epidemiology science along with the clinical microbiology knowledge will help to take various public health measures to stop the outbreak as well as stop the epidemic and pandemic. So to summarize, we have learned what is microbiology as a science. It's a vast subject, and one of its component is medical microbiology, which has various branches like bacteriology, virology, mycology, and parasitology. What is clinical microbiology? Basically, it is a application of medical microbiology for the infectious disease management, and we have seen what are the components and applications of clinical microbiology. Thank you.